observations of the tract structures and their interaction and the rationale behind it, as this is a novel aspect of our nanoscopic approach. In fact, the simulations and the modeling at the atomistic level can help us understand and explain the underlying mechanisms by which molecular radiotherapy achieves its beneficial effects. As an example, I refer to a recent publication explanation of the oxygen deletion mechanism at the flash ultra high dose range using the present methodology.
Yes.
Such understanding is substantial to employ molecular radiotherapy optimally to design experiments, preclinical studies, and clinical trials. I would like to start my introduction with this popular image taken from the planet Earth by Voyager at the edge of the solar system. As we know, our outer space environment is nothing but filled with the radiation, with energies that ranges from 3 Kelvin background radiation to high energy heavy charged particles. Therefore, we may think of Earth as a biological singularity in the universe due to the persistence of the life owing to the protection mechanisms from the atmosphere that blocks most of the hazardous ionizing radiation and protect our DNA from the lethal damage. As we can see in this slide, the scale of the problem ranges from billions of kilometers to sub-nanometer and millions of years to attoseconds, a multi-scale space-time problem. So to start with, here are some fundamental facts we need to consider in radiobiology and radiotherapy. The human body is composed of trillions of cells. Cells provide structure for the body, take in nutrition from food, oxygen from air, and convert those ingredients into energy for specialized functions. The DNA materials in form of chromosomes are localized in the cell nucleus, and any unrepairable damage to the DNA may result in alteration of the cell functionality. DNA is a double helix chain of nucleic acids or nucleotides tagged as T, A, C, and G, which is abbreviations for thymine, adenine, cytosine, and guanine. In the central dogma of molecular biology, DNA contains instructions or coding that are used to create proteins. The pathway starts with the DNA replication and transcription, that is a mechanism for construction of a messenger RNA in cell nucleus. The translation of genetic codes to synthesize proteins takes place in cytoplasm by transfer RNA, which carry amino acids to the ribosomes. These amino acids are building blocks of proteins. Therefore, any interruption to this protein synthesis machinery may lead in alteration of the cell functionality. For example, suppression of the gene P53 causes carcinogenesis, that is, irregular cell division with no checkpoints and stops. It's important to know not all damages may alter the DNA genetic codes because not all of the genetic sequences present within a DNA molecule actually code for the proteins. In fact, less than 2% of our DNA codes are for genetic shape-up, and the rest, which is 98%, are simply junk DNA. Classification of the DNA damaged by ionizing radiation is the following. It includes the damage to the base that holds the integrity of the DNA double helix structure by the hydrogen bonds in addition to the genetic makeup, or damage to the backbone in form of single strand break. A critical damage to DNA is double strand break, which is a pair of single strand breaks within 10 base pairs. For example, one single strand break in the left backbone and another single strand break in the right backbone within a couple of base pairs can be considered as a double strand break. The purpose of this talk is to review the role of nano-dosimetry and the effect of ionizing radiation at the molecular level on the treatment of the cancer. So let me start with a review of the cancer and treatment modalities developed over the last decades. Cancer or carcinogenesis is characterized by uncontrolled division of the cells that form a tumor. Thus, the cancerous tissues undergo abnormal growth. In addition, their ability in invading other normal organs distinguishes cancerous cells from normal cells, a mechanism known as metastasis that occurs through the circulation system and accumulates cancerous cells at the lymph nodes. The level of invasive cancerous cells is potentially related to their surface morphology, and some people have claimed correlations with the fractal dimensionalities and geometries. In any case, if the spread is not controlled, it results in death and mortality. The treatment of the cancer is very challenging. There are various modalities developed for treatment of cancer, such as surgery, chemotherapy, radiation therapy, hormone therapy, gene therapy, and finally immunotherapy. In radiation therapy, which is the focus of this talk, at the earliest stage of the disease for well-localized tumors, a curative radiation dose is prescribed. And the expectation is that there would be a longer lifetime for the patients. But cancer can spread out through vessels and lymphatic system, and metastasis may take place in advanced stages of the disease, in which it is challenging or even impossible to control the disease. As an example, metastatic tumors in lung may invade unrelated tissues, such as in brain. And if metastasis spreads over the various areas of the brain, a whole brain radiotherapy may be the only option of the treatment, in which a loss of cognitive ability might be a result of that. A recent, I'm hearing some background noise. What is the source of that? Maybe
yeah,
some people need to mute their microphone.
Yeah, please, audience, please have your mic muted.
Yeah. Thank you. So a recent study on mouse shows that the application of ultra-high dose rates of radiation, known as flash maybe.
Ramin, I think your mic has got muted, so could you please unmute and continue?
Can you hear me?
Yes.
Okay. To examine occurrence of hypothetical mechanisms at flash dose rates, such as oxygen depletion, we propose a approach at the nanoscale and simulated formation of non-reactive oxygen species from interacting reactive oxygen species. So such exotic models as we proposed at the atomistic level would have practical applications in medical physics. And this is the topic of this present talk. Based on this hypothesis, normal tissues are rendered hypoxic and become radioresistant, and thus they spur. But because tumors are generally hypoxic and being unaffected by the dose rate, but application of high dose rates preserves and maintain the immune system to fight against the tumor cells. Therefore, the application of flash dose rate is ultimately beneficial to suppress faster, even for the hypoxic tumors. Okay. To understand the workflow and clinical aspects of the radiotherapy, let me consider the typical workflow in diagnosis of prostate cancer and the follow-up procedures. This is one example. As a first step, typically physicians use the scores of prostate-specific antigen or PSA and Gleason from biopsy. If the score of Gleason would be above a critical value, either surgery or typical external beam therapy with, for example, 180 centigrade per fraction for 44 to 45 fractions, which is each fraction happen in a single day, will be prescribed. Noting that the long-term effects of radiotherapy, such as 5 to 15 years efficacy and survival rate, have been shown to be equivalent of the surgery. In these lines, there are few jargons, including gray, that is the SI unit of the dose, which is the deposition of 1 joule of ionization radiation in 1 kilogram of tissue. That are used. We should note that the ionizing radiation is any type of radiation that generates ions or a local in tissue that changes the chemical environment of the cell with no heating mechanisms as a cause of the cell damage. Example of this, the source of this ionizing radiation is for 6 to 21 megavolt of photons or 80 to 250 mega electron volts protons. Here is a sketch of workflow in a typical radiotherapy center. First, patients undergo a CT scan that simulates the treatment positioning and assign a system of coordinates to the digital images. The images will be exported to a treatment planning system for reconstruction and registration of the patient data that includes the anatomy of the patient and the tumor. The images will be uploaded to a computer-based software that allows the physicians and other staff to go through the patient's anatomy and identify the location of the disease and the tumor. From this step, typically a dosimetrist or a physicist add a margin to the target, contours the normal organs, calculate the dose, and optimize the plan. The optimization process includes conforming and maximizing the dose coverage to a specific target and minimizing of the exposure to the normal tissue and organs. Then another physicists go through the work done by the dosimetrist and review the plan. He or she conducts a series of measurements known as patient QA, which is quality assurance, to make sure there is no any issue regarding to the computerized workflow that controls the. And once the plan is signed off by the MD and the physicist, it would be ready for the patient treatment. The rest of the treatment is done by the therapist on a daily basis routine, which a challenge to repeat the process each day with 1 to 2 millimeter tolerance in patient's position. And we should be aware that several techniques have been developed to maintain the mechanical uncertainties in patient positioning within such tiny limits. To understand the aspects of a typical treatment machine in external beam therapy, let's take a look at a video that I uploaded from the Electa, which is a company which makes medical linear accelerators, and this video is also available on YouTube. So let's start and take a look at it.
I think the audio is off from the video. If you can explain or maybe turn on the audio.
Can you hear the video?
Yeah, but not the video audio. Not the audio from the video.
So you didn't have a sound on here?
No.
Not to this point?
No. Okay, no sound till now.
Okay, I thought we fixed that, but...
Yeah, I think when you do the present again and include audio, that will be it.
Okay.
So if you stop the present and try it again.
All right, hold on. My audio is on. I'm not sure.
It's the computer audio you have to include in your present.
Yeah.
And try present again with.
Oh, okay. All right.
The audio.
Okay.
How the linear
Now we can
Now
hear
we
the
can
sound.
hear the sound.
Okay.
How the linear accelerator works. All body cells can be damaged or killed by radiation, but tumor cells are more sensitive to radiation than normal cells. Radiation therapy uses this principle to damage beyond repair or kill the abnormal cancer cells in a tumor. Successful radiation therapy depends on the ability of the linear accelerator, or LINAC, to deliver a tumoricidal dose of radiation to the tumor, while ensuring minimal radiation of normal tissue. The LINAC accurately produces, monitors, controls, and conforms the radiation beam to the planned target. Beam generation. Radio frequency waves are pulsed into the waveguide by the magnetron. This is synchronized with the injection of electrons into the waveguide by the electron gun. The radio frequency waves accelerate the electrons along the waveguide to a speed approaching the speed of light. The X-ray beam is created when the electrons hit and interact with a tungsten target at the opposite end. The magnetron controls the power and frequency of the radio frequency waves, which determine the energy of the X-rays produced. The digital accelerator uses a diode-type electron gun situated at the end of the waveguide. The electrons are produced by heating the tungsten filament within the cathode and are then injected into the waveguide. The number of electrons injected is controlled by the temperature of the filament. The electrons are accelerated along the waveguide toward the target. The waveguide contains a series of copper cells. Small holes, or irises, between these copper cells allow the electrons to travel along the waveguide and help to focus the beam. A vacuum is created to ensure that the electron beam is not impeded by other particles. The path of the negatively charged electron beam is controlled by two sets of quadrupole magnets, called steering coils, that surround the waveguide. An additional two sets of focusing coils help to further define the electron beam so that it is very fine, with a diameter similar to that of a pinhead when it hits the target. The entire system is cooled by water. The electrons exit the waveguide and enter the flight tube, where the beam is redirected towards the target. The electrons travel along a slalom path within the flight tube. 3 pairs of magnets on either side of the flight tube cause the electron beam to bend through the turns of the slalom. This process not only positions the beam to strike the target, but it also further focuses the beam to a diameter of 1 millimeter. The design of the magnets enables them to focus electrons of slightly different energies onto the same point on the target. This is called achromatic behavior. This slalom bending is unique to electrolinear accelerators. It helps to minimize the size of the machine and ensures that its isocenter remains low, which is important for patient setup. The high-energy electrons hit a small tungsten target where the electron energy is converted into photons, or X-rays. The high-energy photons emerge from the target in a variety of directions. The primary collimator only allows forward-traveling X-rays to pass through, creating a cone-shaped beam. The primary collimator minimizes leakage, and therefore excess total body dose, by absorbing scattered X-rays traveling in the lateral direction. It also defines the maximum size of the resulting clinical radiation beam. At this stage, the photons are not uniformly distributed across the beam, and so a flattening filter is placed in the path of the beam. The cone-shaped filter absorbs more photons from the center of the beam than from the sides, creating a uniform photon beam. Dose measurement. The photons now pass through the ion chamber for dose measuring and beam quality monitoring. the dose delivered to the patient is measured and controlled simultaneously in two independent ionization chambers. One chamber is the primary dosimeter. It measures the radiation and terminates the beam when the required dose has been delivered. The secondary ion chamber acts as a backup and will stop the irradiation if the primary chamber fails. The treatment machine must replicate the beams modeled within the planning system. This is critical to the accuracy of treatment delivery. A beam quality function is performed by a third ionization chamber, which uses 7 electrodes to monitor different sections of the radiation field. The X-ray beam is almost ready to treat the patient. Before that, further beam shaping is required to ensure that the shape of the delivered X-ray beam matches the shape of the tumor. This is done using a multi-leaf collimator, number of fine tungsten leaves which move independently of one another and can create a variety of complex treatment shapes. LINAC control. One computer system controls both the LINAC and the multi-leaf collimator. This eliminates dosimetric errors due to communication delays. It also ensures synchronization between the delivered dose and the multi-leaf collimator position, allowing complex deliveries such as intensity-modulated radiation therapy and volumetric modulated arc therapy. All electromagnets, steering, and focusing coils are digitally controlled. All mechanical positions of flight tube, filters, and foils are automatically selected from the control console, and all radiation beam settings are grouped in calibration blocks for each energy. These are stored digitally on the LINAC hard disk for flexible and easy beam adjustment, calibration, and servicing. Clearance. Clearance is the free space available under the LINAC for patient treatment, and it varies with different protocols and fixation devices. It is a combination of the distance between the lower surface of the radiation head and the isocenter, 45 centimeters, and the head diameter, 62 centimeters. A wide clearance around the isocenter means improved access for patient setup, freedom to use the best possible patient positioning and immobilization accessories, freedom to rotate the gantry between fields without needing to move the patient. Finally, it means that treatment techniques using non-coplanar beams are not compromised. The large clearance offered by Elekta machines ensures flexibility in providing the best possible treatment for the patient.
Okay. Can you hear me?
Yes. Yes.
Okay.
So,
Let me see if I should. Okay. Now in this video clip that I prepared by myself, which I'm going to show, various aspects of the computerized planning system will be shown. This is an example of a prostate treatment planning system. So this software is known as Eclipse and developed by the company Varian, that is the major manufacturer of the medical linear accelerators in US. And it is a part of a larger package that starts from this CAT scan and ends up with the linear accelerator control system, as we saw in the previous video. Using these computerized applications, the physicians enter their prescription in this case, a daily dose of 180 centigrade in 44 days, with total dose of 7920 centigrade. The images that you can see in this video are in the transversal, coronal, and sagittal views of the patient body scanned by the CT. The combination of these planar images produce a 3D image of a patient anatomy, and the grayscale of the images that you can see were calculated from the measurement of the Huntsville unit by the CAT scan. That is a measure of the tissue density. So we can distinguish, for example, bone from soft tissue, fat, air, and etc. In addition, a 3D view of the patient is simulated by the software. In this plan, the dose was already calculated through an optimization approach that maximizes the dose coverage to the target and minimizes the exposure to the normal tissue. This can be visualized by a 2D projection of a 3D dose distribution in each plane, as you can see in this video. In addition to a plot of the volume histogram for each organ. Also, the dose distribution corresponding to the clinical influence is shown. This is the map used for the verification of validation of the plan, namely comparing the calculation versus the measurement in procedure known as patient QA. So here you can see different organs, which is the bladder, prostate, and femoral heads, and bones, some parts of the bowel, and etc. So that, for example, the yellow is bladder, that green was prostate. Now we are going to see the dose distribution. So these are the isodose lines, and here is the color plot of the dose. This is 100% coverage, which we see that the entire prostate is covered by the dose prescribed by the physician. But still, there are some parts of the other organs, such as bladder or other rectum or other tissues, which are covered by the radiation. And that's what we want to minimize. So we want to maximize the coverage of the prostate by the radiation and minimize the exposure to other organs. And here is a histogram of the exposure to every tissue, such as bladder or rectum. And so we want to, we use this as a measure of if you wish, toxicity to these organs. Okay, let me end this video and switch to, I guess I'm running behind the schedule, but Imran, you can stop me whenever I exceed that 1 hour limit. So as we saw, optimization problem in radiotherapy consists of maximizing the dose coverage to the target and minimizing the exposure to the normal tissue. The aim of radiotherapy is to target the malignant cells and break their DNA. However, the induction of a lesion in DNA is incorporated by a very complex enzymatic repair mechanism. In addition, the radiation exposure to T cells and lymphocytes and suppresses the patient's immune system locally or through the entire body if a whole body radiation was used. Therefore, we are not only deal with an optimization in space domains, but we are also dealing with optimization in time domain. So this is practically an optimization problem in a space and time. In that regard, the dose rate is a major factor that can be considered as a tuning variable. The dose rate is an important factor to compete against the DNA repair and preserve the immune systems to be able to fight against the tumor cells. And again, the efficacy of flash dose rate, for example, in suppressing malignant cells might be explained by this hypothetical scenario of less damage to the immune cells. So these are the things that I'm going to show through the rest of my talk. At the cellular level, the success and failure of the cell lethality is a stochastic problem that can be formulated in a binary distribution function that is a continuous function of the exposure dose known in the literature as TCP and NTCP, which are the abbreviation for tumor control probability and normal tissue complication probability. And in case of heavy particles, TCP and NTCP are proposed to be a continuous function of the particle linear energy transfer, or LAT. And that's the topic of this paper we published recently. And in this paper, we use a pool of patients treated for ependymoma in MD Anderson in Texas, and we showed that for a given probability of the toxicity, as LAT increases, the dose decreases. And the entire formulation, again, is based on this multi-scale model we propose, which is the details of this formulation can be found in other paper published by the same group in 2019. So the details of multi-scaling approach and the steps in computing TCP and NTCP starting from atomistic scale was presented in this article, in which DSPs and DNA damage at the atomistic scales lead to the cell survival at the macroscopic scale, which is measurable experimentally. So we use that formulations to fit the experimental data. This slide covers the same general ideas that I pointed out in my previous slide. The movie in the right corner is an animation of the DNA damage and DSP formations after deposition of energy and ionization of DNA environment by a track of proton and formation of the OH free radicals. As you can see in the lower part of the DNA, the chemicals induced by radiation form single and double strand breaks, whereas the upper part of the DNA, a double helix structure that did not go through a radiation exposure, remains intact and no damage is scored. In general, the radiobiological events starts from attosecond times and continue in a cascade of cellular responses to days and years. Technically, to cover the entire spectrum of the events in space and time domains, we not only must develop appropriate models at the specific scales, but the most computational challenge is matching the boundaries between different domains of nano, macro, and macro scales. I should note that although the DNA damage is kind of the central dogma of the radiobiology, but there are other responses that might be explained through the damages to the other biological pathways and disruption of communication channels, cell signaling, and immune systems. And the model that I present here can be extended to study such classes of the effects. Okay, so some of our centers are using the proton therapy, and I showed in the previous slide that I simulated the track of the proton. So it is important to know the difference between the therapy by photons and protons. And I prepared another video. It's a kind of a shorter video to show the difference and what the proton therapy is about. I hope you have a sound, and if you don't, please let me know.
Today, there are three main types of treatment to fight cancer: surgery, chemotherapy, and radiotherapy. Unlike traditional radiotherapy that uses photons to irradiate and kill tumor cells, proton therapy is an advanced form of treatment using protons. These particles give proton therapy its many advantages. Proton therapy's greatest advantage over conventional radiation therapy is its accuracy and its ability to spare healthy tissues. Conventional X-rays are made of photons that pass through the body and deposit considerable energy before and after the tumor. Protons, however, have a particular physical property called the Bragg peak. They're able to deposit less energy on their way to the tumor and do not affect tissues beyond it. This property allows to confine the radiation dose to the tumor and radically decreases the risk of injury to surrounding healthy tissues. The proton originates from the iron source, where hydrogen atoms are separated into electrons and protons. The protons are injected into the cyclotron, where they're accelerated. Then, these high-power protons are sent through an energy selection system and a degrader that adjust their energy. The beam transport system conducts the protons with the right energy and trajectory. Then the proton beam arrives in the treatment room via a gantry. The gantry revolves 360 degrees around the patient so that the beam can be delivered at any angle. A high accuracy of patient positioning is required. IBA's treatment rooms are equipped with the latest patient positioners, alignment, and imaging systems to ensure the reproducibility of patient positioning. Today, proton therapy patients have access to a state-of-the-art delivery mode called pencil beam scanning. The beam is steered across the target volume one layer at a time, pixel by pixel, to precisely match the shape of the tumor. With this active scanning technique, the physicians are able to change the proton dose at any specific location in the target. This allows proton therapy to adapt to very complex shaped or moving tumors, improves the dose conformity, and reduces the integral dose of radiation given to the patient. Proton therapy is changing the way you treat cancer.
Did you hear the voice?
Yes.
Yes.
Okay, all right. So with regards to the concept of the time, and in particular the dose rate, it is also important to know that the different cells have different replication cycles. But some cells at some stage of our life do not undergo regeneration. For example, mature neurons are in G0 phase, meaning no cell regeneration happens. So any damage to mature neurons by excessive radiation causes a permanent damage, ranges from loss of memory or a severe damage to nerve systems that may even lead to the death. Some cells, including stem cells, have 4 stages of cell cycle. And the topological structure of a DNA molecule changes as a function of time based on the cell cycle interval. So the radiosensitivity of the DNA and cell itself changes as a function of time. The most radiosensitive stage of a cell is in the mitosis, where the DNA is unpacked and ready to divide as shown in this video. I prepared another video to give us what is the mitosis and what is the stage of the cell cycle.
Mitosis begins as the long threads of DNA in the nucleus start to coil. Having already replicated, these threads emerge as the double strands we know as chromosomes. Meanwhile, protein fibers grow from the migrating centrioles, forming a latticework of spindles. For reasons scientists don't fully understand, the nuclear membrane suddenly disintegrates. With amazing accuracy, a spindle from each centriole attaches itself to each of the chromosomes. Assisted by the protein fibers, the chromosomes move center stage. In a microscopic tug of war, the spindles pull each chromatid toward opposite poles. Other spindles push against each other in a ratcheting action that stretches the cell. The chromosomes then unwind and the nuclear membranes reform. With mitosis complete, the nuclei and cytoplasm separate, creating two new cells in this ongoing cycle that sustains all life.
So if any DNA damage happened at this stage, and if the chromosomes are broken apart, and if some part of the broken chromosome goes to one cell and other one goes to another cell, then a chromosome aberration may transfer to the next generation of the cells. And so the other aspect of the time and dose rate is related to DNA repair mechanism. The 2015 Nobel Prize in Chemistry was given to the discovery of a DNA repair pathways followed by ionizing radiation. So as we learned so far, the DNA double-strand break are the most dangerous type of a DNA lesion because they can potentially propagate to the next generation of the cells, as we observed in the previous slide. However, only a small fractions are lethal because of the repair mechanism. In fact, cells deal with the DNA damage throughout various mechanisms, including homologous and non-homologous enjoining. That includes handling of roughly 50 double-strand breaks per cell cycle. The video that I'm going to show gives you a flavor of homologous enjoining pathway that requires having a replica of a DNA to be used as a template for the repair mechanisms to take place. So let's take a look at this video and
1 million women all over the world suffer from breast cancer. A healthy breast consists of different cell types. In the nucleus of the cells, you can find the DNA. Every day, the DNA of each cell is damaged thousands of times. If only one strand of the DNA is damaged, this can be easily repaired. PARP proteins bind to the damage and attract other proteins that repair the damage. When damaged DNA duplicates before the damage is repaired, a double-strand break can arise. In a healthy cell, this more severe damage can be repaired with the help of homologous recombination. BRCA1 protein binds the loose ends of the DNA and starts to unravel the DNA strands. Using the undamaged DNA strand as a template, the damage is then repaired. Finally, the two strands are separated, leaving both DNA strands error-free. In patients with hereditary breast cancer, the BRCA1 mechanism does not work. When a double-strand break arises in the cells of these patients, the DNA cannot be repaired. The persisting damage can cause cells to die, but it can also transform a cell into a cancer cell. This cancer cell is the start of a tumor in which all cells have the same defect.
Okay, so so it is important to know the exposure of radiation is not in synchrony with the cell cycle, as we saw in the last two videos. And because of the random distribution of cell cycles among the cells in a tissue, many damages occur with no available DNA templates for the homologous end joining mechanisms, as we saw in the last video. Alternatively, a second pathway in cell, so-called homologous end joining, will be triggered. This repair process is prone to error and may lead to a chromosome aberrations and permanent mutations in genetic codes, and this may cause a number of the abnormalities. So here is a list of potential damages that may take place in fetus, as an example, if excessive exposure to man-made radiations happen. We should note that a large radiosensitive stem cells from the rapid growth of cells in fetus are prone to the DNA damage. That might lead to various types of late cancers, such as leukemia, brain cancer, and other chromosome aberrations, growth abnormalities, such as mental retardations, microcephaly, and growth retardation. Okay. So I may also need like 10 more minutes. Do I have that time, or?
Yes.
Yes.
Yeah, sure. Yes.
Yeah, okay. So now we want to make a model of all of these things that we talk about. And essentially, this is a very complex problem. So in order to do the modeling, the simplest way is to do Monte Carlo. And in order to do the Monte Carlo, we have to understand the nature of the interaction of the ionizing radiation with the matter or tissue. As we saw, a common modality in radiotherapy is based on usage of a megavoltage generated photons, which are uncharged particles. So the list of the interactions they can cause with the tissue varies from photoelectric Compton scattering, electron per positron per production, and nuclear reactions. As we see in this diagram, a release of charged particles occurs as a result of photon interaction. So, for example, the electrons are released in photoelectric or Compton scattering. These secondary charged particles travel up to a few centimeters in tissue, and along their path, they interact constantly with the valence electrons and deposit their energy. So, in a nutshell, this process changes the local chemistry of the tissue by induction of a local plasma. And that's the source of the DNA damage, as we discussed. In case of charged particles, if we consider a continuous slowing down approximation, particles deposit energy to matter according to the Bethe formula. And this is the equation for the energy deposition. According to this formula, the charged particle's velocity in the denominator of the stopping power diverges as very low energies, making the relative deposited energy into this shape, with a peak known as the Bragg peak. And this is a peak discussed in one of the videos I showed for the proton therapy. A major advantage of using charged particles, as we discussed, such as in protons, would be their finite range. So they cannot go beyond the range of the range of the proton, which is going to have zero velocity. But in case of the photon, because photon carries no rest mass, they penetrate and there is no limit to stop a photon as long as it exists. So here in the next video, I prepare a track structure of the photons. So here the green lines are the photons. The red lines are electrons, and blue lines are protons. In the middle of this video, you see a box, which is a tissue, equivalent matter, for example, water. And in the middle, you can see a sphere, which is with the density of the gold. So this is a gold nanoparticle. I chose gold nanoparticle to increase the photon scattering cross sections due to the high density of the gold relative to the water. And as we can see in this video, photons rarely interact with gold nanoparticles, and many of them pass through the cells without a single interaction. And this is a hallmark and character of uncharged particles interacting with matter. But upon their interactions, they generate cascades of charged and uncharged particles, including photons, neutrons, in addition to a large number of secondary electrons or delta rays through Compton scattering, photoelectric effects, and preproductions. So the charged particles deposit their energy along their path, literally in a continuous form. The yellow dots here are ionization points. So we can see as we send more and more photons, there would be a distributions of ions within the cell nucleus or cell in general. Okay. In the next video, I show the track structures of the electrons, and you can see the difference. The electrons are in red. As shown, all electrons interact with cell from entrance in addition to gold nanoparticle in a discrete set of ionizations. And because of the relatively light mass of electrons, they are prone to a strong lateral scattering. So their tracks diverges right after electrons enter to the tissue. And there is a small overlap among the ionization points among different tracks. And this is a character of the electrons. And the next video shows the track structure of the proton. And because protons carry a mass 2,000 times more than the mass of electrons, they have much less lateral scattering. The process of ionization in proton versus electron is slow because the compactness of ionization that makes the proton to be biologically more damaging. And the overlap among the particle tracks is substantially higher compared to the electrons. There are a couple of remarks on the simulation of the dose rate in Monte Carlo, problem with, for example, the flash that I should mention. First, to the best of my knowledge, there is no implementation of the dose rate in any Monte Carlo toolbox. In the toolbox I use in these videos, which is GN4 or GN4 DNA and Topaz. And the the extensions of the topaz, which is n by n. In this Monte Carlo toolbox, the tracks are simulated independently without relative time lapse and with no interaction among their chemicals. And all of these things must be added manually outside of this toolbox. So in order to simulate the dose rate, for example, we have to bring the different particles in a sequence of time. We can adjust on the tracks relative to natural time scales of the molecules, namely their time scales associated with their vibrational or excitational modes that are within 1 femtosecond. For example, consider if we send 2 tracks sequentially with a time gap corresponding to 10 cycles of the molecular vibrations. And if one cycle accounts for 1 femtosecond, then the time gap between 2 tracks would be 10 femtoseconds. In this example, if we tend to increase the dose rate by a factor of 2, we simply send the second track after 5 vibrations. So we have to play with this Monte Carlo toolbox to bring the concept of the time and the dose rate for our simulations. And we did it using molecular dynamics, like in fact. So the time scale associated with different events are summarized in this chart, which go from earliest stage of radiation at a second up to days and years. It is indeed a highly challenging problem to cover the entire range of these time scales with a single computational model, and I'm not aware of any computational tool that covers the details of the events within entire range of these time series. So as mentioned previously, we may employ different models developed for specific scales and patch them together. In our approach, starting from the attosecond events, we use GM4 DNA Monte Carlo and score the coordinates of ionization and excitations in space embedded by molecular dynamics simulation bonds. As the ionizations and excitations may alter the chemical bonds, one needs to take into account the structural instabilities induced by direct ionization. And this calculation can be done through quantum mechanical simulations such as Carr-Parinello molecular dynamics, or VASP, which was developed originally for chemical reactions or solid state or condensed matter physics. As we identify the initial structure of the molecules using ab initio calculation, we can simulate the evolution of the molecules and their reaction diffusion by another type of molecular dynamics known as react force rate that starts from fraction of femtosecond up to 1 picosecond and 1 nanosecond at most. And subsequent to that, we use a microscopic coarse-grained model to lump large number of degrees of freedom into few variables. And that model is useful for the time evolution of the chemicals up to microsecond and beyond. So this is the concept of the multi-scale modeling that we use in time domain. All right. Now, when we send ionizing radiation to environment of the DNA, some toxic chemicals such as free radicals can be generated. Here in this, I prepared another video which shows what the free radicals do to molecules such as guanine, which is the base of the DNA, or sugar moiety of DNA, which is part of the DNA backbone. Here, if you can see, there is an isolated red and white ball, which is a OH free radical. Oxygen represented by the red and hydrogen by the white ball. And we can see here in this video that oxygen grabs one hydrogen and become a water molecules, and the DNA loses one hydrogen. So this is a simple damage. The same thing happened for the guanine, in which a guanine loses one hydrogen and free radicals becomes a water molecule. These OH radicals can interact. They can produce hydrogen peroxides. I'm not going to go through this. Other things that we can propose by doing these simulations is by flipping or pumping spin to a chunk of the DNA, which the idea is coming from spintronics. I'm pretty sure Imran remembers those days back in Buffalo, where we used to work on some interesting ideas of spintronics. So if we can inject one spin to the molecules as the way that we did in those solid state materials, then we can manipulate the radioresistivity of this molecule. So as we can see, even a free radical exists around the DNA molecule, it cannot react. And the simple idea is, you can see in the left corner of this slide, in which there is a magnetic repulsion between free radicals and the DNA. And this magnetic repulsion coming because the free radical has an unpaired spin with spin one hand. So if the DNA molecule carries a similar spin multiplicity, then there would be a repulsive force. Here, this video shows the effect of the environment. Because I don't have enough time, I'm going to skip. But the important aspects of this video is the hydrogen bonds among water molecules and free radicals, in which it produces a viscosity or some dragging force among these chemical species. Here is simulations of single-strand breaks and double-strand breaks, and we saw these movies before, and so I'm going to skip them again. We don't have enough time. These are interesting videos in which it shows if we have a very high dose of radiation, in case of the flash, then we have reactions among OH radicals, and they form non-reactive species. And this non-reactive species have a large mass and low diffusibility, and therefore they are less reactive. And so based on these molecular simulations at nanoscale, we propose rate equations in which N1 here in these rate equations represents the density of the reactive oxygen species, or ROS, and N2 represents the density of the non-reactive species, which do not interact with the DNA. And the right-hand side of this, G represents the dose rate, N1 squared represents the interaction among the ROS, N2 and N1, the third term in the equation 1, represents the interaction between NROS and ROS in formation and formation more massive non-reactive species. And the second equation is the generation of NROS because of the interaction among the reactive species. And analytical solutions of these equations can be given. It shows that N1 linearly scales by the dose rate, and N2 quadratically scales by the dose rate. Therefore, if we increase the dose rate dramatically, N2, which is the number of the non-reactive oxygens, are going to increase, and they become the dominant. So that's the point. We realize that this is the effect of the flash, in which it has less toxicity to the tissue than because of oxygen depletion, and this is a mechanisms we develop to explain the oxygen depletion. And so I would like again to emphasize these rate equations are based on the molecular simulations we did at the nanoscale, and so we proposed this explanation, and we were able to fit a qualitative picture of the flash and compare it with the conventional dose rates in which the oxygen, if the oxygen depletion is a hypothetical mechanism, then this is the model that we propose, and apparently this is kind of a satisfactory model in which it explains different aspects of oxygen depletion. Okay, so because I don't have enough time, I jump to the results and conclusions. We presented the first principle molecular dynamics simulations and obtained a mechanism for oxygen depletion hypothesis through the nanoscopic and macroscopic scales and patching their boundaries. This mechanism explains the normal tissue injury observed in flash dose rate, and so the time-dependent molecular simulation revealed the formation of a non-reactive oxygen species, in which explains the reason why the normal tissue can be spared at the flash dose rates. So let me wrap it up and jump to this acknowledgement, and let's open the session for any question. Thank you.
Thank you,
Dr. Ramin. If there are some questions, so we invite questions. Any questions? Yes, Dr. Manvi. I think that was a very interesting talk.
Hello, Dr. Sunil, Imran, Dr. Imran. Am I audible?
Yes, go ahead.
Okay. My question is to Dr. Ramin. First of all, thank you, Dr. Ramin, to deliver an impressive talk. Myself, Dr. Manveer, and my question is that which technique used during the radiation therapy to measure the exposure radiation dose, especially in proton therapy, which you discuss in your impressive talk.
Sorry, can you repeat your question one more time?
Yes. My question is that which technique used during the radiation therapy to measure the exposure radiation dose?
Oh, we did Monte Carlo simulations, and through these Monte Carlo simulations, we are able to calculate the dose, and that's from the calculation.
Okay, it means that this is the theoretical method to measure the exposure radiation dose by Monte Carlo method. But at a particular site in our body, the tissues, neighbor tissue, how can we measure the absorption dose by the tissues?
Well, that's coming from the stopping power, and that the method formula I showed you. So this is a basic for equation in which it shows as the charged particle go through a tissue, there is an continuous energy transfer to the tissue, and that energy, if you divide that energy to the mass of the deposition area, then we can calculate the dose in unit of degree. So, and this has been done through a Monte Carlo simulation.
I think if I understood, Dr. Manveer wants to know, like during the treatment, is there any experimental way to get those, external doses to the?
Yes, that is a process that we call it patient QA. I show it in one of my movies, in which let me go back to. So here is. So this is if you see in the left corner of this of this box, I have something which is written prostate, and below that it is other plug which is a QA. So that QA was created for this patient. This is photon, this is not proton. But the same concept applies to the proton. The only difference is the type of calculation, in which for the proton, MCNP or any Monte Carlo package can be used to calculate the dose. Here, the algorithm for the calculation of the dose in photon for the variant machines or Eclipse is, for example, AAA or other method, which are kind of superposition convolution method. So here, let me go. Oh yeah. So in the previous part of this movie, you saw a human body. Now you can see here other thing, which is like a cylinder, and the cylinder is filled with water and a distribution of the ion chambers and diodes. Those ion chambers and diodes does the measurement of the dose, and then we experimentally verify the calculation of the dose using this device, which is designed for the rapid arc. So this is a measurement which verify or validate the calculation for the patient. So did I answer your question? Or so for each patient we do this measurement and validation.
Okay, Dr. Ramin, thank you.
Okay.
Thank you. Any other question? If not, then I invite Dr. K.K. Gupta. He is the teacher in charge of the Physics department from this college for the vote of thanks. Dr. K.K. Gupta, over to you.
Hello, Dr. Sunil. Am I audible?
Yes, sir.
Yes, sir.
Professor Ramin, Abolfath, our principal, Professor Manoj Khanna, our colleagues, students, and all the participants. It gives me great honor and privilege to deliver the vote of thanks at this event. I would like to thank our chief guests, Professor Ramin Abolfath, for his wonderful talk and inspirational talk on the path of computational nano-dosimetry in medical physics. Hope this talk will inspire our young generation, young students to enter in this field where we are lacking in our college, medical physics. Particularly the medical physics, we don't have much facilities here. I think Dr. Sunil, Dr. Imran will have now enough work in this field. I would also like to thank our principal, Professor Manoj Khanna, Dr. Sunil Kumar, Dr. Imran Khan for organizing this event, Dr. Hemant Kumar Rajor, IQSC convener, all my colleagues, all students, and Mr. Rahul Jain, and all the participants present here through the Google Meet for making this event a grand success. Once again, I thank you all present here through the Google Meet. Thank you.
Thank you very much.
Thank you very much, sir. Let us end this meeting. Thank you very much once again to follow.
Thank you, Ramin, and thanks for taking the time. It must be very late. So, I think it's better to close it, and the recording would be available for the students. If they have any questions, I will give your email so they can also ask you that.
Thank you very much, Imran. See you soon.
Okay, see you. Take care. Bye.
Good night.